Hello, students. So today we are going to talk about chapter 15, the chromosomal basis of inheritance. So if we analyze this chapter states as to whether it is the chromosomes or DNA that is the actual basis of inheritance. So it was Thomas Hunt Morgan who worked on the Drosophila melanogaster. Drosophila is the genus, melanogaster is the species of the fruit fly. Now, why did he take fruit flies to study his genetics experiments? Firstly, because fruit flies, they produce many offspring at a time. They breed every 2 weeks, and they have only 4 pairs of chromosomes, a total of 8 chromosomes. Now, Morgan researched on 2 types of fruit flies. He worked on the normal and the wild type. So the normal or the wild type, they were the common ones in the fly populations. Those traits that were alternative to the normal or the wild type were called the mutant phenotypes. So the first mutant Morgan discovered was a fly with white eyes instead of the normal wild type red-eyed flies. So we'll look at the slide which shows the wild type red-eyed. So the red-eyed were the normal fruit flies. The normal fruit fly color was red eyes, and the mutant variety were the white-eyed fruit flies. So in one experiment, he mated male flies that had white eyes, which were the mutant variety, with female flies that had the red eyes, which were the normal wild type variety. And as you saw in the previous chapter in Mendelian genetics, we worked on monohybrid crosses, and we saw that the ratio in a monohybrid cross is always 3:1. So how did he prove this 3:1 cross in his genetic experiment being conducted with the fruit flies? So in the fruit fly generation, in the F1, which is the first filial generation, all the fruit flies, they had red eyes. And in the second generation, they had a 3:1 red to white eye ratio. That means 3 of the fruit flies, they had red eyes, and only 1 had the mutant white-eyed. But who had the white eyes were only the males. The females had the red, normal, wild-type red eyes. So Morgan reasoned that the white-eyed mutant allele had to have been located on the X chromosome. So the males that carried the mutant white-eyed color was on account of the X chromosome. So Morgan's finding supported the chromosome theory of inheritance. Humans and other mammals, if you look at our chromosomes, which we learned in our karyotype, we humans have 23 pairs of chromosomes or 46 chromosomes. 22 pairs of our chromosomes were called autosomes, and we all possess only 1 sex pair of chromosome. If I am female, I have the XX chromosome, and the males would have XY chromosome. In the same way, what Morgan proved was that those flies that were white-eyed, which carried the mutant variety, were the males, and they happened to be on the X chromosome. So we see here that always the Y chromosome in humans determines the sex, the male sex, and it is shorter than the X chromosome. Our X chromosomes are always longer. And so this is the test cross that Morgan carried out. I encourage you to practice this cross. In the first generation, we saw when he took red-eyed females, which were the normal wild type, and he crossed it with the white-eyed males. All the offspring in the F1 generation had red eyes. When he took the F1 generation and once again crossed them in the second filial generation, sons and daughters, what he obtained, as I just mentioned above, was that 3 were red eyes and only 1 mutant white eyes. But the 2 females were the red eyes. 1 male had red eyes, but the other male had the recessive white eye, which was carried on the X chromosome. And so if you carry out over here, if you see the wild type, which was on the X chromosome, and you see the female had the red eyes and the white eyes were on the X chromosome, but this was the white-eyed mutant variety. When he crossed it, the eggs and the sperm, what he saw was he received the female with the red eye, which was the normal wild type, and the male had the mutant variety. And that's what you can obtain in the cross. And finally, in the F2 generation, we saw over here that in the F1, all had red eyes. In the F2 generation, Thomas Hunt Morgan received 2 females with red eyes, 1 male with red eyes, but only 1 male with the mutant white type variety. So now he compared different systems and what were the inheritance amongst insects, humans, the rooster, the cock, and what was the haplodiploid system. This is a very important slide. You will receive many questions. You could be asked questions on this slide. So we just talked about the XY system, which is the system present in humans. We know we have 44 pairs of autosomes and just 1 pair of sex chromosomes. So males have the Y chromosomes, so they have X and Y, and females, we have the XX chromosome. That is what we refer to as the XY system present in humans. Let's go to the next system, which you see in grasshoppers. It's called the exosystem. So we see in the exosystem that they have 23 pairs again. They have 22 and 1 pair, which is 23, which is 22. The males would have 1 X, and that's why they have a missing, which is called the exosystem, and the females will have XX. So 22 plus XX. Here we have 44 plus XY, which makes it 46. You have 22, 23, 24. So total number in the grasshopper would be 24 chromosomes. Let's look at the rooster, the cock, or the cocks. What they have is what we call the ZW system. Who has this ZW system would be the females. They have 76 plus total 78 chromosomes. So the females will have ZW, whereas the males will have ZZ. It's just the opposite if you want to think. Males in us, humans, have XY. Here, their males will have the same type of chromosome ZZ, and the females will have the W. If you look at the haplodiploid, we know we've talked of haplodiploid. In meiosis or sexual reproduction in humans, we have what we call the haploid gametes or the sex cells and the diploid parent cell. So the haplodiploid would be, they have 32. The females have double the number of chromosomes as that of the males. So they have the diploid number, and the males will have the haploid, which is only 16 chromosomes. So what we notice is during meiosis in males, The male chromosome, which is the Y chromosome, is always shorter and will carry fewer number of genes. And the short segments at the ends of these Y chromosomes, they are homologous with the X, allowing the two to behave like homologs during meiosis in males. Now, because of the genes that are present on the Y chromosome, those genes are referred to as SRY genes, SRY, which stands for sex determining region on the Y chromosome, which is responsible for the development of testes in an embryo in males, and a gene that is located on the X chromosome is called the X-linked chromosome. The genes located on the Y chromosome are called the Y-linked genes or the X-linked genes. And a gene that is located on either the X or the Y chromosome are referred to as sex-linked genes. So genes on the X chromosome are called X-linked genes, and the human X chromosome contains about 1,000 to 100 genes. The X chromosome have genes for many characteristics that are not related to sex. Many Y-linked genes are related to sex. And X-linked genes are going to follow a specific pattern of inheritance. For a recessive X-linked trait to be expressed, a female needs to have 2 copies of the allele. Therefore, the female is said to be homozygous. A male will only have 1 X chromosome, that is one copy of the allele. Therefore, the male is said to be hemizygous. So X-linked recessive disorders are much more common in males than in females. Some of the disorders that are carried by the X chromosome in humans is caused by the recessive alleles present on the X chromosome, and therefore the diseases that are linked to the X chromosome or the X-linked diseases are color blindness, Duchenne muscular dystrophy, and hemophilia. Now, each chromosome we've seen has hundreds or thousands of genes except the Y chromosome. Genes that are located on the same chromosome tend to be inherited together, and therefore they are called linked genes. So how does linkage now affect inheritance? So again, Morgan conducted experiments with fruit flies to see how the linkage is going to affect the inheritance of 2 characteristics or 2 traits. Morgan crossed flies that differed in traits of body color and wing size. So the first cross was a P generation cross to generate FY. So he carried a cross where he was wanting to see 2 traits. He looked at the body color and he looked at wing size. And the second was a test cross that he carried out. So when he found that body color and wing size are actually inherited together in specific combinations, and those genes that do not assort independently were on the same chromosomes. As I mentioned earlier, this is the chromosome and these are all genes together. So genes that are close together on the chromosome are inherited together. Genes that are located further away on the chromosome will be inherited later. And that's what Morgan determined while carrying out his genetic experiments, that these genes do not assort independently. And therefore, he reasoned that they had to be on the same chromosome. So this is exactly what we would carry out in the dihybrid cross, where we took 2 pairs of contrasting traits, which Gregor Mendel carried out. He looked at yellow round seeds and green wrinkled seeds. So he looked at the color of the seed and he looked at the shape of the seed. And what he got was 16 different combinations, because 4 times 4 would be 16 different offsprings that he carried out. in the test cross that he conducted. Now, when we looked at meiosis in prophase 1, I particularly stated that there were many processes like crossing over of the homologous chromosomes that took place in prophase 1. And because of crossing over of the homologous chromosomes, we saw genetic information was exchanged here. At this intersection point, the X-shaped region, which we call chiasma, where synapses occurred. And so we can clearly see the crossing over is resulting in new combinations of genes or traits. So recombinant chromosomes bring alleles together in new combinations in gametes. Think about it. If crossing over did not occur, all of us would be having the same traits, inheriting the same traits. It is on account of crossing over that we have different combinations of alleles in the sex cells, which are the gametes. So random fertilization is also increasing the number of different variations that can be produced in a sex cell or gamete. And this abundance of the genetic information is the raw material on which natural selection is going to work. This involves genetic recombination, the production. What is genetic recombination? You're recombining the genes. It's the production of the offspring with combination of traits differing from either parent. So if you notice, some of us, you may have inherited a trait that neither of your parents possess. Because remember, a gene is a unit of hereditary information that is being transmitted from parent to offspring for generation to generation. And so you may have inherited a trait from your great-great-grandparents, either from your maternal mother's side or from your paternal side. And this is on account of the genetic recombination that has occurred. So you could have completely new combination of genes which determine our traits. So now what we want to see, what are the different ways in which the recombination data can be mapped. And so Alfred Sturtevant, one of Morgan's students, he constructed a genetic map. What is a genetic map? It is an ordered list of the genetic loci. What is a loci? It is the location or the position of a gene on a chromosome. So we've seen that there are hundreds of thousands of genes on a chromosome. And geneticists, as I mentioned earlier, do not know the exact number of genes present on a chromosome. So a linkage map is a genetic map of a chromosome that is based on recombination frequencies. And map units indicate the relative distance and the order, but it cannot indicate the precise locations of the genes. They're only indicating the relative distance and the order in which the gene is present on the chromosome. And as we saw, genes that are further apart on the same chromosome, they have a recombination frequency of 50% because they would be inherited together. Those that are located close to each other will be inherited together. So such genes are physically linked, but genetically they are unlinked. And therefore they behave as if they were found on different chromosomes. We've seen a number of abnormal chromosome number, which led to a number of genetic diseases like in Down syndrome, or genetic disorders like in Down syndrome, where the individual had 47 chromosomes. They had one extra copy of the chromosome, and that was due to a process called non-disjunctions, where the extra copy of chromosome failed to separate out from its pair on chromosome number 21. And as a result, one gamete received 1 two of the same type of chromosomes and another gamete would receive only one copy. Now, in aneuploidy, what results is, it results as on account of the fertilization of the gametes in which non-disjunction occurred. So aneuploidy was seen in Down syndrome. Offspring with this condition have the abnormal number of a particular chromosome that we saw, 47. Monosomic, one zygote has only one copy of a particular chromosome. Trisomic, as we saw in Down syndrome, has 3 copies of a particular chromosome. So we can see in meiosis 1, all this occurred because of non-disjunction, failure of this extra chromosome to separate out from its pair in meiosis 1. And in meiosis 2, as we can see here, this non-disjunction occurred because it failed to separate out from the pair. So we talked of aneuploidy. Now we look at polyploidy. Poly means many. So it is a condition in which an organism has more than 2 complete sets of chromosomes. And in polyploidy, you could have 3 sets of chromosomes, you could have 4 sets of chromosomes. So if there are 3 sets of chromosomes per cell, then it's called triploidy. Tetra comes from the word 4, so it's 4 and 4 sets of chromosomes, not 4 chromosomes, 4 sets or pairs of chromosomes. Now, where do we find polyploidy? Polyploidy is mostly seen or is most common in plants, but not in animals. So in animals, we see aneuploidy and not polyploidy as in Down syndrome. So these words are extremely important. Aneuploidy. It's on account of this non-disjunction, failure of that extra copy of chromosome to separate out from its pair. Whereas polyploidy is typically seen in plants and not animals. So now, because of all these aberrations or these disruptions that have occurred on the chromosome, it is resulting in all these genetic disorders. So what are these alterations in the chromosome structure that can occur? Breakage of chromosome can lead to 4 different types of changes in chromosome structure. One called deletion, duplication, inversion, or translocation. We're going to see in the next slide exactly how deletion, translocation, inversion, or duplication is taking place. So let's analyze these terms. Delete. To delete is to remove. So deletion is the removal of a chromosomal fragment, and part of a chromosome is removed. A fragment of the chromosome is removed. Think about it, students. When we are looking at fragments of chromosome, what is chromosome? Chromosome is a rod-shaped structure that contains the genetic information, DNA. So if part of your DNA of the chromosome, which contains your genetic information, is removed, that is an aberration. It's a mistake leading to a disease. Duplication. When to duplicate something is to copy itself. So the sequence is being repeated, that segment of the chromosome that contains the genetic information is repeated. Inversion. Inversion is to reverse the orientation of a segment within a chromosome. So if this is my finger, this is my whole chromosome. So all the genes that are present here now, if I invert it, if I call this for understanding purposes, this is A, this is B, end of the chromosome. Now I've inverted it. So now B comes on top, A comes down. What has happened is the genetic information that was here is reversed. And remember, genes specify our traits. So we're going to see how inversion is going to affect. Translocation is you're moving a segment from one chromosome to another chromosome. So these are all the alterations that occur. So let's go with deletion. So if you have chromosome here, which is represented by different segments, A, B, C, D, E, F, G, H. And now, if the letter D, this part of the chromosome is removed, firstly, it is going to shorten the chromosome. Part of that information is lost. And so you have only A, B, C, E, F, G, H. D is missing. Let's go to duplication, A, B, C, D, E, F, G, H. Now in this chromosome structure, you can see the letter B and C, those segments are repeated. So you have A, B, C, D, E, F, G, H. So this makes the chromosome longer with repetitive segment of the chromosome information. Let's go to inversion. So you have A, B, C, D, E, F, G, H. Now, B and D fragments are switching places. They're inverting themselves. And so now you have A, D, C, B, E, F, G, H. And what happens, because B has reversed to D, taken D's place and D has taken B's place, all those genes that were present in these two locations now have been inverted. And so the traits that they are going to specify are going to be different. Translocation. You have two chromosomes here, A through H and M, N, O, P, Q, R. Now, A and B are switching places. Part of the segment here is moved to this segment on the other chromosome, and this chromosome moves positions. So now you get M and O, C, D, E, F, G, H, and you have A, B, P, Q, R. This becomes shorter and this chromosome becomes longer. So it moves a segment from one chromosome to a non-homologous chromosome. So we've talked about these genetic disorders in meiosis, all about Down syndrome. And we saw clearly trisomy 21 due to non-disjunction, it's aneuploidy. And this is just to reiterate the information. This was the karyotype, 22 pairs of autosomes and that one pair of sex chromosome. But we notice that this individual here on the 21st pair, instead of having a normal pair of chromosomes, has this extra, which is trisomy condition. And this is the individual who suffers from Down syndrome. Aneuploidy, as I mentioned to you, non-disjunction of sex chromosomes produces a variety of aneuploid conditions. We talked about Klinefelter's, which was XXY. We talked about Turner syndrome, which was exochromosome. And you could also have XXX, as I mentioned to you, but didn't go into details. XXX would be the females who, and this frequency occurs 1 in every 1,000 females that are born. They are healthy, but with no unusual physical features, though they are at risk of learning disabilities, facing learning disabilities. Now, there is this syndrome, disorders that are caused by structurally altered chromosomes. So what do we mean by structurally altered chromosomes? The chromosome structure has been changed. That one type of particular syndrome is referred to cri du chat, French, which means cry of the cat. And it results from a specific deletion. So what is one segment on chromosome number 5 is deleted. And the child that is born with the syndrome is severely intellectually disabled, impaired or disabled, and has a cat-like cry. And individuals are going to usually die at infancy or in early childhood. Now, certain cancers, including chronic myelogenous leukemia, CML, are also caused because of translocation. Remember, we just earlier talked of translocation when a segment of the chromosome or a part of a chromosome is going to move to another chromosome. And that is these two. Now let's see firsthand what happens in translocation. So you have a normal chromosome number 9 here, and you have normal chromosome number 22. But part of this chromosome on segment on chromosome number 9 and chromosome number 22 are going to switch position. So you see here, chromosome number 9 is reciprocal translocation. What do we mean by reciprocal translocation? So part of chromosome number 9 switches and goes to chromosome number, part of chromosome 22 switches position and goes to chromosome number 9. And part of 9 comes on to 22. So they are reciprocating with each other in terms of its genetic information. Translocated chromosome 22 is now referred to as Philadelphia chromosome. Let's see, what do we mean by genomic imprinting? For a few mammalian traits, the phenotype is going to depend on which parent passed along the alleles for those traits. Such variation in phenotype is called genomic imprinting, which involves the silencing of certain genes depending on which parent passes them on. So that is referred to as genomic imprinting. The variation in phenotype is genomic imprinting because part of the genes, well, certain genes will be silenced. And that depends on which of the parents, whether the male or the female has passed on these genes. So students, I encourage you, go through each of these concepts. We looked at the genetic disorders, study all these genetic disorders. They are important, and as I had given you 2 sets of review questions for the final exams. You will be able to answer those questions once you have reviewed these chapters.